Well, it's my pleasure today to introduce Dr. Hinman. So Dr. Hinman is a professor and physician scientist in the Department of Neurology at UCLA, as well as the interim director for the stroke service at the West Los Angeles VA Medical Center. He received his MD and Ph.D. from the Boston University School of Medicine in 2007, and he did his residency training at UCLA before becoming faculty. He's received many awards, including the top neurologist in 2017 from the International Association for Healthcare Professionals, as well as Southern California Super Doctors Rising Star Award, which sounds awesome, that same year. His research focuses on utilizing and developing tools for cell-specific transcriptional profiling in models of neurological disease. And this research is funded by several grants from the National Institute of Neurological Disorders and Stroke, as well as the American Heart Association COVID Rapid Response. On behalf of the organizers of the Brain Bee, it's my pleasure to welcome you today, Dr. Hinman, and we really look forward to your presentation.
Thanks, Dr. Cahill. It's really a pleasure to be here. I hadn't really known about the Brain Bee. I had heard about it, but I hadn't really known about it. And I'm really delighted to be here and share some of our work. And you asked me to talk specifically about COVID-19 and its effects on the brain. And so we've since the whole scientific community has been relatively paralyzed by COVID-19, we decided to jump into it, and I'm hopeful to share today with you some of the background and some of the information that's out there in the scientific and medical literature about the relationship between COVID-19 infections and the brain, and then hopefully some of our interesting data. So let me make sure I can share the screen.
Yep, it's all there. We're good.
Okay, great. Let me move my toolbar. There we go. So yeah, so as Dr. Cahill said, I'm a physician scientist. I'm privileged to have the opportunity both to see patients at the bedside and evaluate them. My practice is mainly focused on stroke, but I'm a neurologist, see all kinds of interesting cases, and then really be able to take some of the things that we don't really know about the brain and study them in the laboratory. And so it's really, it's been a long training pathway to get there, as Dr. Cahill indicated, but it's really one of the most, in my opinion, one of the most rewarding career paths that's out there. Let me make sure we can go forward. So, the talk again is about the relationship between COVID-19 and brain disease. This is not really what I intended to spend most of my career working on. We're an interesting lab that focuses on, as Dr. Cahill suggested, understanding, using really modern neuroscience tools to understand the relationship between stroke and Alzheimer's disease, which are two of the most common neurologic diseases out there. And we use all kinds of different tools to ask and answer interesting questions. So that ranges from molecular biology, bioengineering, and genetic analysis, to really using all kinds of different models like cells in culture, even brain organoids, animal models, and human subjects, to really try to understand disease process and then design new treatments for that. And so I'm going to show you some data that later on in the talk that uses some of these tools in the relationship to understanding COVID-19 and brain disease. So we were really excited at the beginning of 2020. We had just gotten some new grants and some new support, and really excited to dive into a whole bunch of interesting experiments. And this is where we work, is the Neuroscience Research Building on the campus of UCLA. And we're up on the 4th floor towards the corner there. And, everybody was really keen to start working, and then this happened. And so we all had to sort of slow down and take a break and make sure that we weren't part of the widespread communication of COVID-19. And this is obviously, we're not alone in this. This is a huge worldwide problem. This is a map which you can access online from Johns Hopkins University. This is a map of active cases of coronavirus as of a few days ago. As you can see, it's still a tremendous worldwide problem, as most are keenly aware. And during that pause where, back in the spring, where everyone was sort of slowed down and asked to think about different things, my group started to think about whether there was any way we could apply our tools and our experimental designs to understanding a relationship that became apparent early on, that COVID-19 was not only a respiratory pneumonia, but also affected the brain. And so I'm going to try to take you through that data and how we came to that, and then show you some experiments that we've done to get at that. But first, I thought, for this audience, when I asked around, like, what would be the most important thing to start off with, most of the younger folks that I spoke to said, well, I don't totally know what coronavirus is even. And so I thought it'd be interesting to tell you a little bit about that. So, and then I'll also say here that throughout my talk, I've embedded some of the really interesting scientific art that's been happening over the last 9 months that was really stimulated and provoked by coronavirus. And so I try to put some of that in throughout the talk. So it's not just words and things to look at, but there's some really interesting scientific art out there that you can look at if you're so inclined. So, coronavirus, or the virus that causes COVID-19, is also known as SARS-CoV-2. That stands for severe acute respiratory syndrome. The kind of pneumonia, the pattern of pneumonia that we often see in severe cases, associated with coronavirus. And it's actually not the first time we met this virus. SARS-CoV-2 is a 30,000 base single-stranded RNA virus. It's a member of a family of coronaviruses that cause a variety of disease, most commonly the common cold. There are 7 others that we know in the family that can cause disease in humans. And of course, this virus first discovered in Wuhan, China, a place I'm sure most of the audience never heard of before this past year. And to me, one of the most remarkable things about the SARS-CoV-2 is that from the moment it was first found to be causing disease in humans somewhere in mid-November of 2019, about 2.5 months later, the full 30,000 base genome was published by Chinese scientists on January 20th. You can still look up that. We know it's changing now, but the original genome is published just 2.5 months after the first human case was found. And that's just really a tremendous scientific achievement that can't be understated. This is what it looks like. It's 30,000 bases, as I mentioned. It doesn't really have a lot of protein. So most of the group hopefully knows that, RNA gets translated from its genetic sequence into proteins. And a virus like this one doesn't have a lot of proteins in it. This is a simplistic view. I'll show you a more detailed view later. But of course, the most famous protein here is this spike protein against which all the vaccines are made. And again, keeping with the scientific art discussions, there's been all kinds of interesting things that have happened. This is a soundtrack of turning that RNA sequence into sound. Hopefully you can hear that. And so, when we think about what we understand about this virus, we have to recognize that a lot of the proteins that you see pictured here are in fact common to RNA viruses. But what's really unique is that spike protein. As I mentioned, it's not the first time we learned about a coronavirus. Probably before many of you were born, we struggled through another worldwide. It was more of an endemic rather than a pandemic, meaning that, epidemic rather than a pandemic, because as you can see from this map, of SARS-CoV-1 infections, the original SARS virus back in 2003, many countries were spared. We did have some cases here in America and in Canada. There were a few in, there was at least one in Europe. It was mainly initiated or found somewhere in China and spread locally in that community. There were only about 8,000 infections documented. That contrast that with the nearly 70 million infections of SARS-CoV-2 that are now around the world. But it's not the first time we knew about this coronavirus. And if you stretch back further, I'll make an interesting argument that we're starting to think about. There was a Russian flu pandemic. Many have heard about the press related to the most recent pandemic, the influenza pandemic of 1918. But 30 years prior to that, there was another worldwide pandemic, which appears to have had its origins in Eastern Russia, and that spread around the world. It's been attributed to influenza, but in point of fact, this is let me just play this video here. This is the spread of, let's see if it'll play, maybe not, that's fine. In point of fact, there's some evidence that perhaps this Russian flu pandemic was in fact the first time that the world met coronavirus. This is a map on the left of the family of coronaviruses. This is from a paper from 2005. You can see SARS-CoV-1 hanging out here. It doesn't have its number because it was all by itself at that time. And only a few of these coronaviruses cause human disease, mainly labeled by the H here. But there's some evidence that this family here with bovine coronavirus, and in particular this human coronavirus OC43, that using a technique called molecular clock, in which you can estimate when a virus may have jumped from animals to humans, there's some suggestion that perhaps this virus, the OC43, the coronavirus that they were studying, in fact, moved from animals to humans around late 1800s, just before the turn of the century. And that happens to correspond importantly to two events. One, a worldwide euthanasia of cattle that were developing not only respiratory symptoms, but neurologic complications that forced them to be put down. It was only 5 years after that that the Russian flu pandemic took hold of the world. And that had an interesting component of neurologic disease that suggests there may be a lot of similarities to what we've been seeing with not only SARS-CoV-1, but now COVID-19. And so there may be a long history of coronaviruses infecting humans and causing not only respiratory disease, but also neurologic illness. Oops. Oh, so yeah, so the next point in the talk here is to mention about what coronavirus does to the body. And so again, some of that interesting scientific art around COVID-19 to show you. One of the things coronavirus does is cause this pneumonia. And this is two ways of looking at pneumonia. On the right, we have a chest x-ray. On the left, we have a CT scan of the more advanced radiographic study to look at the infection. But you can see that it causes this sort of florid pneumonia that makes it difficult for folks to breathe and causes robust inflammatory infection in the body. Now, one of the things that the lungs do that's important is that it reoxygenates the blood. And so while you look at this and you say, oh, there's a lot of air moving through the lungs, there's also a lot of blood moving through the lungs. So as we've learned now, as we've taken care of COVID-19 patients, it's not only a respiratory pneumonia, but it also has a component of a bloodstream disease in which there's a lot of inflammation and thrombotic, meaning clot formation, that's associated with this disease. As we started to see many more cases spreading around the world, we now recognize that this is not only a respiratory pneumonia, but it affects all other types of organs. There's measurable incidence of kidney failure. There's some suggestion that various forms of heart disease are in fact associated with COVID-19. As I mentioned, we've been seeing patients who get lots of blood clots in their lower extremities and also in their lungs. We see this interesting kind of rash that can develop on the body in some cases within the skin. And then there's been a variety of neurologic complaints that include headaches, dizziness, confusion. Another term for confusion can be encephalopathy, muscle aches, and loss of taste and smell, of course, are two of the most common complaints. And then there's a measurable increase in the risk of stroke associated with COVID-19, and our research really is focused on that. And so just now we'll get into some of those complications that we attribute to the relationship of COVID-19 in the brain. I just put a quote here from the literature. This is one of the earlier reports of from the BBC of a gentleman who had suffered multiple strokes associated with his COVID-19 infection while living in the UK. And the most salient quote from the scientists and physicians in that article is that from early May is that it's quite clear that the virus does in fact cause problems in the brain. I know there's been some debate and some discussion about that, but I can endorse that there definitely is a clear association. And again, it's important to think about the history here. And I'm again not a virologist, nor, as this slide indicates, a veterinarian. But in my discussions with some virologists, it's really important. The cats actually already know about this. There's such a condition, many who have pets may know about this, feline enteric coronavirus. This is one of the most prevalent infectious diseases in cats and typically causes a robust diarrheal syndrome, an infectious peritonitis or an inflammation of the lining of the gut. But interestingly, 30% of cats with feline enteric coronavirus also have neurologic symptoms. I'm showing here a couple of pictures of what that looks like. This is a portion of the cat brain, a brain stem shown here. And this kind of sometimes the lining of the brain and the brain stem, referred to by the arrow here, can become inflamed. And if that can get into the brain tissue, we refer to that as encephalitis. And here again, a picture of the actual virus in the lining around the brain. And so this is something that the cats can actually tell us quite a bit about through the study of the pathology of their brain and an understanding of aspects of feline enteric coronavirus that might be similar to SARS-CoV-2. They're important genetic differences, but they also have this spike protein, which makes coronaviruses unique in their viral biology. We've seen cases of encephalitis in COVID-19. Here are some radiographic examples. These are a couple of folks. This is a brain MRI study, so it uses magnetism to understand the amount of water in brain tissue, and we can use that but through mathematical transformations to understand how different structures of the brain and skull are what they look like, and then how they might be damaged by certain disease processes. And in general, the brain should be back here, this sort of homogeneous gray with the cerebral cortex being able to be differentiated from the underlying white matter, bundles of axons that connect the brain together. And really, there should be no bright white signal. That generally indicates that water and inflammation are leaking out into the brain, which is not a normal process, but a diseased one. This is a 58-year-old flight attendant who had a 3-day history of cough, fever, and mental status changes. And she had evidence of SARS-CoV-2 in her nostrils. And she ultimately developed this kind of radiographic change in the MRI showing this fluffy inflammatory changes in the deep portions of the brain called the thalamus, and then also in her temporal lobes in a region of the brain referred to as the hippocampus. And so this can cause confusion, disorganized thinking, and in some cases, seizure. Here's another example of a 26-year-old who again developed seizure phenomenon. In this case, he didn't have a nasal swab that was positive for COVID-19, but rather his spinal fluid demonstrated evidence of the infection. And this is, again, a diagnosis of encephalitis in the brain. So there definitely is a small group of patients in which the virus can get into the brain. We don't totally understand how, perhaps through the bloodstream, across the blood-brain barrier, or perhaps through an organ in the brain called the choroid plexus that makes spinal fluid. That may be another important mode of transmission. There's also a suggestion that it could come through the nose, through the olfactory nerve that lies right the top of your nose, or basically right here, and transmits smells into the brain. And of course, that's a proximity effect for the virus, which is hanging out in the nose and the airways, and maybe using the olfactory nerve to get into the brain directly. The other, sort of really popular, at least in the lay press, popular neurologic symptom associated with COVID is this post-COVID fog, brain fog that people sometimes refer to. The real definition of this means that someone's having neurologic symptoms that occur after their COVID-19 infection. So they might have cleared the infection, but they're still having cognitive complaints. They may be feeling fatigue throughout their body. They may have a sensation of dizziness, even though they're sitting still or resting. Headaches are quite common. There have been a few cases of even psychosis associated with this. And we really don't understand this process at all. It's an active area of research, and there are multiple different explanations. One could be changes in the blood-brain barrier that impact the ability of causing sort of a low-grade inflammation, maybe a very, very mild encephalitis, like you might attribute to the prior slides that I showed. The other is that when you get a lot of inflammation in the body, sometimes those inflammatory molecules can find their way into the brain and disrupt synaptic transmission. And so that could be a reasonable explanation for the kinds of memory complaints and confusion that people have documented. Again, this is not the first time we've experienced this. After the 1918 influenza pandemic, there were nearly 5 million cases of encephalitis lethargica. This is a visual representation from an artist at the time. And this is, again, a post-viral syndrome. It occurred after the respiratory flu, in which folks were developing some of the very same symptoms that we're seeing now in this post-COVID brain fog: confusion, in rare cases, psychosis. And in fact, that was associated with a kind of a movement disorder that made folks very somewhat still and slow to move. And so we still occasionally see this after influenza, but when you see all the cases at once, it tends to make a more robust mark on the medical literature. So we're anticipating that there may be a tremendous outpouring of these post-COVID patients in the coming years and decade. Now, I want to spend the rest of the time talking about what we've been doing our work on, and that's the neurovascular complications associated with COVID-19, which have been one of the more prominent and newsworthy aspects of the infection, the virus. I'll say that what I mean when I say neurovascular complications is generally stroke, but stroke is a basket neurologic term, meaning that there's a sudden change in neurologic status from which there can be multiple causes. And I'm sure I tried to show some pictures here of what those could be. One is that you can get these very tiny strokes. We call them microvascular strokes in the very small blood vessels in the brain, and they appear. They're somewhat difficult to see, and sometimes cause symptoms and sometimes don't, or cause symptoms that are not necessarily dramatic. We've definitely seen an increase in this what we call large vessel stroke, meaning the large blood vessels in the neck and at the base of the skull that supply a lot of blood to the brain that can become occluded. Here's an example of that where the blood flow drops and we get a stroke affecting a significant portion of the brain, causing neurologic symptoms. We've also seen bleeding events in the brain, a condition we call intracerebral hemorrhage, where blood vessel ruptures and blood is sort of exposed or transmitted into the brain, and that disrupts neurologic communication because it's not supposed to be there. We've also seen this condition, which is called venous thrombosis. So when you think about strokes, you only think about the arteries that go to the brain, but all those arteries ultimately lead to veins that drain all that blood, and they can become clotted too. This is a relatively minor portion of most of the strokes that happen, but we've seen a significant increase in venous thrombosis and in cases associated with COVID-19. The blood doesn't drain out properly, you get changes in brain pressure, and that can ultimately lead to a hemorrhage, but more often causes headache and other neurologic symptoms, including changes in vision. For those who are interested, I'll just do a quick shameless plug that I did a podcast a few weeks ago about this, in which I talked about some of these varieties. So if you want more information, you can look for this podcast called the Strokecast. This is a map, and there's been an effort to sort of organize these. This is a global effort. There's now going to be an NIH US-based effort to understand all the neurologic complications associated with COVID-19. And this is a graph that shows when we sort of expect some of those things to occur, when we've seen them occur. And so at the top here, you have diagnostics showing that, you get nasal swab and then potentially antibodies several weeks later after the infection. The second portion of the graph here in orange shows the types of clinical symptoms that one might expect. And first, off you have the sort of common COVID-19 symptoms of fatigue, fever, cough, maybe headache, maybe muscle aches. When you have encephalitis, it's generally been documented in the early phases. We've had some cases of demyelinating disease, meaning the loss of the wrapping around axons, both in the brain and in the peripheral nerves. Although that association I didn't include in the talk because the literature are somewhat fuzzy on that. The best recent study says maybe that's not happening. And then strokes, which we've seen both early on, sometimes even in the absence of COVID-19 respiratory symptoms, as late as several weeks after the COVID-19 infection. And so I think it's still, this disease remains somewhat of a mystery. And as I said on that podcast, I'll reiterate here, it's easy to forget, we're good at taking care of patients, but it's easy to forget that before November of last year, no doctor on the planet had ever seen this disease before. So it's evolving and we're still learning about what it can and can't do, and certainly how to treat it in its best way. So as we go through then the really understanding the relationship between COVID-19 and stroke, I'm going to take you through several geographic areas which helped inform our understanding. And this is an interesting map that I found, which shows how COVID-19 actually spread around the world through air travel. So here's the center in Wuhan in China. That's the airport in Wuhan. And then you can see that a series of worldwide flights made an important contribution to the spread and pattern of COVID-19 around the world. And so we'll start in Wuhan because our Chinese colleagues documented very well some of the disease processes that were happening there. We'll then jump over to Italy. From Italy, we'll jump to the UK, across to New York, and then ultimately to Los Angeles, and we can talk about some of our work. So it was first in Wuhan where it was discovered that in addition to the respiratory pneumonia, there were brain events and neurologic symptoms that were associated with this disease. And the Chinese, in the very first reports from the main hospital in Wuhan, described a 3.5% rate of those infected with COVID-19 having neurologic symptoms. That doesn't sound like a lot necessarily, but if you then extrapolate that there are 70 million plus cases now, we're talking about a lot of folks who have been affected by this. And if you had a severe form of COVID-19, you were much more likely to have a neurologic outcome. In fact, you were about 6.5 times more likely if you were in an older age group to both have a severe form of COVID-19 and also have neurologic disease. And this figure here represents probably the seminal association of the CT scan that I showed earlier showing evidence of COVID-19 in a patient who also had a stroke, but presented first with the stroke symptoms, not the respiratory symptoms of COVID-19. And so this is when we really started to understand that there might be this strange association between the two pathologies. Let's go to Italy for a moment. And in Lombardy, Italy, which many remember was dramatically affected by the virus and the pandemic, really the first Western descriptions of what we might be facing in the coming year. There they saw a high rate of comorbid COVID-19 and stroke, nearly 20%. This is a graph in the upper right here showing the types of admissions that the hospital would typically get for neurologic complaints, including stroke. You can see in the blue line, they tend to fluctuate over the course of the spring. Some weeks it's up, some weeks it's down. You can see that once COVID-19 hit in late February, early March, there was a significant increase that then, they associated with, clearly has to be related to COVID-19 because their normal admissions were not really changing too much. And importantly, about 60% of those admitted with a neurologic diagnosis during that period had stroke as their primary complaint. And they often also had a worse outcome. This is a graph, just you can look at the bottom here, the number of patients who come in with a stroke and leave the hospital with what we consider to be a good outcome, meaning some of the paralysis or symptoms they had have resolved. And only 25% of those patients who had COVID-19 did achieve that good outcome, whereas nearly 70% who didn't have COVID-19 were able to recover. So it was not only causing more strokes, but also worse strokes in the Italian population. In the UK, they organized pretty rapidly to try to understand this. It's one of the advantages of having a uniform health system. But they were able to rapidly organize all the most of the neurologists into different regions of the UK to start reporting the association of neurologic disease with the respiratory illness. And you can see from this graph that they found a very disturbing increase with nearly 20 and 30% rates in those over 60 of cerebrovascular disease. But they also documented strokes in patients under 50, which is something we almost never see. And so in such a short period, it would be extremely rare to have strokes occurring in those in their 20s, 30s, and 40s. And so you can also see here from the graph the association of the so-called neuropsychiatric symptoms, a little bit like the brain fog that I was talking about. You can follow this group, the COVID-19 working group. You can follow them on Twitter. They have a relatively updated map of where neurologic complications from COVID-19 are occurring throughout the UK. And they really set the standard for how to track this within a large region. Let's jump over to New York, and many remember that the pandemic hit pretty hard in late March and early April. And despite that, our colleagues there were able to report out what they were seeing and doing despite the impact on hospitals. What they saw, it was an 8.4% rate of stroke in folks who are hospitalized with COVID-19. For some perspective, that's nearly 7 times the rate of a comparable period in which folks sometimes get influenza and have stroke associated with that because of the inflammation. And this was nearly 7 times that. Most of the patients that were coming in were having larger forms of stroke. That's shown in the workflow here. And was reported in the New England Journal and made some press articles with, again, younger folks without COVID-19 symptoms having presenting coming into the hospital with large strokes, a very unusual pattern. What you can see from this graph in the upper right here is that there's really sort of a dichotomy, two populations of folks that have neurovascular disease associated with COVID-19. One is that this group here, the orange dot represents the stroke. And you can see there's this group that came in initially with stroke, no COVID-19 symptoms. Those weren't the presenting reason they came to the hospital. And then you can see there's this other group out here that was having strokes some 10, 20, even 30 days after they were admitted to the hospital or moved to the ICU. And we sort of know about this group. So it's not uncommon to get have more disease the sicker you get. But again, the frequency here is quite high compared to other infectious diseases. And then this population here is definitely very interesting, in which the first presenting symptom of a respiratory pneumonia is in fact a neurologic complication, not a respiratory one. And so, really, there's still a lot of ongoing work to understand whether these are, related to each other, or are they in fact different mechanisms, and we're interested in that. Here's a week in the life at UCLA. This is just a short diversity of cases that we experienced here in Los Angeles in and around Christmas time and the holidays. So just in this short space of a week, we've seen, 2 weeks, we've seen cases like this, a young lady who was caring for her husband. He got very sick with COVID. She wasn't aware that she had it. Her husband went to the hospital. She was coming home from the hospital. She developed stroke symptoms, was taken to the hospital, where we found this occlusive clot in her internal carotid artery, in a usual place. Usually they occur here where the artery splits into two. This is an unusual pattern. She ultimately was diagnosed with COVID-19, but again, not her presenting symptoms. We've seen strokes in the deeper part of the brain. There's a brainstem infarct in an individual who was struggling with COVID-19. Here's another example of a total carotid occlusion. So here's the carotid artery down here on a CT angiogram, and you should see this sort of lighter gray material, and it should go all the way up, as it does on this side, and into the brain. And you can see there's no blood flow on this left side of the brain, and that's concerning. And here's a young person who had a aortic thrombus, something we almost never see in clinical practice, squarely placed right in the aorta as the blood comes out of the heart, and that had broken off into small pieces to cause this pattern of stroke. Again, we've seen those hemorrhages that I talked about. Here's an example of that into the ventricular system of the brain. And then a young man that I took care of, 36 years old, his whole family had gotten COVID. He was the only one who wasn't having a lot of respiratory symptoms and was able to do all the chores and whatnot. And then he developed a headache about a week into his infection. He came down to UCLA having nearly all of the veins clotted off in his brain. This doesn't show it so well without a comparator, but the top vein, the spiritosagittal sinus, was completely occluded. And then all the veins on this left side of the brain were blocked. He was able to get a little bit of blood flow down through the right side. We were able to treat that, and both with by going into the brain, removing the clot, and then also giving him blood thinners. And so he ended up doing quite well. But we've seen the same kinds of patterns that others had described, and we now, I think it's even more perplexing that we see all different manner of neurovascular complications. So when we started to think about how we might look at this in the laboratory, we did what most scientists would do. We sat down, we started reading and see if we could figure out what might be involved in the relationship between this new weird virus and what we know about the way brain blood vessels work and how they regulate themselves. And this is a figure that we put together. Actually, one of my UCLA undergraduate students who was on her way to medical school, but was so eager to participate that she spent her summer working with us over Zoom to organize the literature and understand what might be going on. And so, I mean, she's really dedicated to helping us work on this. And so what we see is that the virus binds to this protein that's expressed in the lung. It's called ACE2, or angiotensin converting enzyme 2. And the only reason we knew this is because SARS-1 also did that. So there were already some scientific evidence that, that this interaction between the spike protein on the surface of the virus might interact with ACE2. And again, we took advantage of what was already known in the literature to really recognize that this might be important. And well, so what happens? Well, when the virus binds to ACE2, it goes inside the cell, and then in something called an endosome, and that is then disrupted, and the RNA from the virus gets out into the cell. It's able to be translated, reproduce itself, and then it goes out. But by manipulating ACE2, one of the things that happens is you disrupt the normal signaling that happens between the molecule, the normal function of what ACE2 is supposed to be doing. And that is to cleave a protein that circulates in the bloodstream called angiotensin 2 into these two peptides that activate receptors on the surface of endothelial cells to do all kinds of good things, like keep blood vessels wide open and relaxed, and then also turn off inflammatory genes. And so when you disrupt that signaling, what happens is you turn on all these inflammatory genes. You also get blood vessels that start to close down. They're not as relaxed as they should be. And all these inflammatory molecules that leak out, they not only go into the bloodstream to exacerbate the ongoing inflammation, but they also act directly on the blood vessels to cause leakage of the blood-brain barrier, to release proteins that we think are implicated in clot formation, like the complement system. And so we started to become interested in whether we had a system that might be useful to understand whether this proposed pathway might in fact be relevant. Interestingly, in that same paper, we wanted to explore the idea, was there any association of the pathways that I just showed you in the figure, and whether there's anything about those pathways that might be already relevant to stroke. And in fact, many of the drugs that were already in clinical trials for COVID-19 listed on the left many of them already had some evidence, usually in animal models, they might, be effective in the treatment of stroke. And so we really decided to that there was some association here and whether we should be looking at some of these things. And so we started with a question that was a little bit unresolved in the early papers on this topic. And that is whether the molecule ACE2 that the virus uses to get into the brain, into the cell, whether in whatever organ you're talking about, whether that was in fact expressed in the brain. And so we did some work taking advantage of some ongoing research at UCLA where we treat patients with epilepsy or seizures. And one of the things you can do to treat that is take out the seizure causing region of the brain. And my colleague had been interested in whether there were changes in endothelial cells between tumors and that normal tissue that we removed from patients. They isolated endothelial cells where you put an antibody tagged to a little magnetic bead, and then you can pull out those interesting cells using a magnet. And they had looked at the expression of genes in those cells. So they already had all the data. We didn't have to do other experiments. This was when UCLA was closed down and there was no research happening. And what we found is that in that normal tissue, in those endothelial cells, we don't really see any of the ACE2. This was pretty consistent with a lot of the existing literature because everybody had been doing the same thing, looking at the information they already had to help understand the disease. And there are very few papers showing that there's any measurable expression of this ACE2. So how does that work? How does the virus get in then? We knew that there was evidence in the lung and in the kidney that the virus does in fact get into the endothelial cells, and showing that here in the bottom. And we knew that that's true in the lung because people were having all kinds of vascular problems in their lung, including clots. So it must be happening. But is it happening in the brain tissue, or is it happening somewhere else? And so we wondered whether there might be an effect of blood flow. This tissue that's resected, it's resected from a living person, but then when you go downstream to look at it, you've lost all the blood flow. So when the time you get to look at the genes many hours after the resection from the actual human brain, the blood flow is gone. And so that is a stimulus for biologic activity in endothelial cells. So then this is when we decided to put our existing models to work. And my colleague, Dr. Naoki Kaneko, is a neurovascular surgeon who has come over to UCLA and been working with us for the last few years. And we had this really neat model that Naoki developed where we can take the imaging studies that we get from patients and they go in to be evaluated for a stroke or an aneurysm, that we get sometimes a picture of their blood vessels. I showed some examples of that earlier. And we can take that, the exact replication of what's generated from the human body, and create a model of it, and then 3D print that model out in the lab to scale. So it's exactly the size of the human blood vessels, and it has all the anatomic features, the twists and turns and abnormalities or normalities that you might see in that particular patient. And this is an example of what that looks like. This is the blood flow pattern or blood vessel pattern from the entire half of the human brain that we were able to replicate. Here's the image generated from the patients. Here's a 3D plastic model that we generate from that. And so with Naoki's paradigm, this is interesting, but it's kind of just an interesting plastic model. How do we make that into an experiment? Well, one thing we can do is we can create a hollowed out model from that plastic. And here's an example. Here we reduce the system just to one segment of the human middle cerebral artery, the main blood flow supply to the brain. This particular example has a narrowing in the middle of the middle cerebral artery, but it represents the exact human anatomy that we gleaned from a patient's study. We can then take human endothelial cells, grow them in a dish in culture, and by putting them into this model with three-dimensional rotation, and then pushing culture media across that continuously, we can create an endothelialized model of that, where the interior surface of the hollowed out model is essentially like a blood vessel. It has the cellular surface that the blood is exposed to. This is a representation of what that looks like. We can study how different proteins are expressed in that model. And so what we did was we took that model and we applied it to this question of whether the blood flow made an important contribution to the expression of the ACE2 molecule that is the entryway for SARS-CoV-2. And what we found is when we put blood flow through the system, you actually increase dramatically the expression of ACE2. Here's an example using both, on the right, using both endothelial cells from the umbilical vein, a sort of just a standard laboratory culture endothelial model, and also then in human brain microvascular cells that line the blood vessels of the brain. And when we add flow, we increase the expression of ACE2, and when we drive the flow even higher, we get even higher expression of ACE2. So the idea is that the flow, the blood flow, is an important variable that might mediate the susceptibility of the virus. So what we did was ask that question. Those were just cells in culture where we can put flow across them just in a sort of horizontal way. We wanted to ask, was that true when we do it in our three-dimensional model that I showed? And in fact, the same is true. So when you have cells grown in the monolayer culture without any flow, there's very little expression of ACE2 shown in the purple. But when when there's a little bit of flow stress shown in the green or a lot of flow stress in the narrowed portion, you increase the amount of ACE2 in the blood vessel. And so this then may be a susceptibility factor for infection of brain blood vessel cells. What we did then was collaborate with another lab at UCLA, Dr. Sung Sai's lab, and that this work was done by a postdoctoral fellow who gotten his PhD already and now is doing extra training as a scientist, Dr. Sandro Sata. And what Sandro made for a separate set of experiments was a liposome. This is like a small circular sphere that has that's made up of phospholipids, which is the same thing as cell membranes made up of. And on the outside of that liposome, he put the viral S protein, which I'm showing here. It ends up looking something like this, which is very similar to what the virus looks like. In fact, the size of the particles was designed to be about the same size as the virus itself, about 100 nanometers. And this is data just showing that S protein liposome does in fact bind to ACE2. When you add ACE2 on top of the liposomes, it gets gobbled up, and you can measure that in a couple different ways. If you add it and then you look for ACE2, it's still bound to the outside of the liposome. And what Sandro did to make this experiment really neat was put a fluorescent molecule inside the liposome. And so when you then look for that's called rhodamine. When you look for the rhodamine inside the liposome, you in fact see that it's there, it's bound to ACE2. So we took Sandro's liposomes and we put them into our model. So this is a microscopy showing the ACE2 level in the 3D model. You can see that it's there. The green just shows all the cells that are present. And when we when we add liposome that doesn't have any S protein on the outside, we don't see any binding to the wall of the blood vessel. But when we flow it through with the S protein on the outside, we see that it binds, it's sticky, and it sits particularly in the regions where we see lots of ACE2 expression, suggesting that flow-mediated increase in ACE2 is really an important factor in the binding or susceptibility of the virus to infecting brain blood vessel cells. What does it do when it gets there? That's an important question, and really speaks to whether there is in fact an association with SARS-CoV-2. And so to do that, we used a rather simplistic experiment where we just took the S protein that normally sits with 3 copies of itself. We call it a trimer. It's all sit together. And we added it to brain blood vessel cells. And then we looked at what happens when you do that. Is there, inflammation that goes up? And one way we do that is to do a technique called direct RNA hybridization, where the RNA in the cell can be measured directly. And what we see when we do that, and I'll draw your attention specifically to panel B here, is that there are these rather distinct molecules, inflammatory molecules, that go up after exposure of brain endothelial cells to the S protein, including one that I mentioned earlier that we're particularly interested in, this complement protein. And so we're working right now to understand that in a little greater detail. I'll just have a few more slides to show you what we're doing with that. This is a table of those slides. This is, again, so the question is the spike protein the only thing that is necessary? Or the whole virus can get in, and what is it doing when it gets there? This is a more complex diagram of the virus. It has this 30 kb genome. The spike protein is right here towards the latter third. Most of the first 2/3 is about replicating the virus, making more copies of itself. And what the interesting thing about SARS viruses is that they have at the end here these genes. They're called open reading frames, meaning that the RNA is in such a way that it's likely to make a protein. But we haven't named that protein because no one's had an opportunity to study it yet in great detail. So they just get numbers like ORF3, ORF7, etc. And there had been some suggestion from the first SARS epidemic that these ORFs might be important. But that went away, and there wasn't really a lot of scientific interest in it. And so the work is it's out there, it suggests things, but it doesn't really show it definitively. So we're left now trying to figure out whether any of these ORFs might play important roles in the clotting that we see. And so what we did in the lab was make a whole series of we call safe viruses that we can use for experiments. They can get into cells. They don't damage them necessarily, but you can put genes in and out, and you can study how those genes then affect the cell biology that you're trying to study. So we've made a whole series of lentiviruses that have each of these small ORF genes that are in the SARS-CoV-2 genome. And I'll show you some really neat data that we just got recently, which is that when you do that and you add that to brain endothelial cells, you get very distinct inflammatory profiles for each of the ORFs. Some of them provoke a very strong transcriptional response like ORF7A and ORF9B. The pattern of increase with each ORF is different depending on the arterial cell type that you look at. So on the right here, we looked at four different endothelial cell types, and the number of unique genes is graphed as the size of the circle. And so you can see that the brain endothelial cells respond differently from the arterial endothelial cells, from endothelial cells in the lung, or that common form that we used in the umbilical vein cells. And this is just a map of the level of gene expression that exists that's different for each individual gene in the different cell types. So we're just getting into this and trying to understand whether these genes within the tail of SARS-CoV-2 might contribute to the neurovascular inflammation that we found triggered by the spike protein. I'll stop there, and I hope there's enough time for questions. I'll just say there's a lot of dedicated scientists who worked throughout the pandemic to help us on this, and they deserve a lot of credit. You know, we really work as a team. This is a photo of the Westwood campus of UCLA about 11 years after the influenza pandemic. It was just getting organized. This is a few, you know, from a few weeks ago. And I think we'll be here another 100 years too when we have to face the next pandemic. Happy to take any questions.
Thanks so much, Jason. So I'm hoping that there may be a few questions, but I'm happy to start it off. I was wondering if you could talk a little bit about whether or not there's big genetic differences in ACE2 expression in people, or, you know, is there evidence that maybe some of these people that had the strokes had susceptibility that might be related to ACE2? Is that a reasonable hypothesis?
Yeah, it is, Dr. Cahill, thanks for asking that question. So, you know, there aren't a lot of known polymorphisms in ACE2. You know, it's not a really well-studied molecule. As I mentioned, it got a lot of popularity and press after the first SARS epidemic, and then sort of drifted off into the background, and there weren't many people working on the system. So I'm sure there are some you know, SNPs that are present, but they're not well characterized. The other thing that makes it difficult is that ACE2 is shed. It's a membrane enzyme. It's shed into the bloodstream. And there is some work to sort of try to measure that and whether that might associate with some of the chronic disease risk factors that may lead to worse COVID outcomes. But, you know, really to know whether you're impacting the function of ACE2, you have to look at those peptides that are cleaved through angiotensin 2. And those are difficult to measure. They're not stable. And so there aren't really good tests for those. So it's been a little bit difficult of a system to sort of scale up rapidly and get a handle on the human disease. I'll also say that another limiting factor here has been that there are really important differences in ACE2 in the human sequence from those in smaller animal models that we might use in the lab, like the common lab mouse or even the rat. And so it's been difficult to develop animal models that can really facilitate work in this area. As you know, many of the vaccines and other things have to be really tested in primates, which is, you know, really a rate limiting step for most scientists.
Yeah, it's really cool though. I love that 3D printer and being able to look at that blood flow. That was really clever. Maybe you can stop sharing screen just for a second and we can see if there's any I see
there's
somebody
a question from,
Tara.
from Tara Lee. If
Yep. You can unmute yourself, Tara, and ask.
Yeah, so hi. Regarding the slide where you had the timeline of the COVID-19 patients versus the acute ischemic stroke diagnosis, I was wondering when you mentioned the young, what range of age that was referring to, and what would be the reason for that range?
Yeah, this is a really perplexing component. Thanks for the question, Tara. You know, it's a really perplexing aspect of the disease because we typically do almost never see strokes in folks under the age of 50, and we've seen a nearly 40% increase in those individuals, sometimes ranging as young as in their late 20s and early 30s, which can be a life-changing event for a lot of people, and we don't understand it. It may be that there are some underlying changes in the brain blood vessels that those folks have that weren't at the stage of causing disease, but are provoked by the infection, or there may be some completely, unusual pathology. I mean, my gut says that most folks at that age have robust cerebral blood flow. They don't have any changes, and as we know, that drives the expression of ACE2, and so that may make them more susceptible. Now, they may also, most people may have good reparative systems, but some may not. And what we may be seeing is that in those folks who can't stop that response to the infection in the endothelial cells, maybe developing the worst types of stroke and other pathologies. Great question.
Thank you.
Chris has his hand up. Chris?
Hey, Jason, thanks very much. So I have a question about how the virus gets into the cells when it binds to angiotensin converting enzyme. Is it internalizing all the time, or are there triggers for the internalization of the enzyme, like other receptors? And
Yeah.
did you know anything about the turnover of the? Is it required that it goes inside the cell for the virus to get in, or can it just bind to the surface? Does anyone know?
So in order to replicate, it has to get in. It has to get in and release its RNA and use the cellular machinery to translate. But as our work shows, just the S protein interaction with the cell surface is enough to disrupt the signaling and potentially trigger inflammatory events. So I don't know it's required to get in order to cause vascular damage. Certainly in order to replicate and move on. The first part of your question, there are cofactors required for that entry. The spike protein exists in this trimer that really makes this bulky head on the S protein, and that has to be cleaved off in order for the virus to get in. It's, there's a metalloprotease called TMPRSS2 that has been implicated in the lung as an important cofactor. Folks are right now in clinical trials studying whether blocking that enzyme can in fact reduce infection, but it's not expressed in the brain. All the single cell data says it's not there. We did 6 or 7 experiments at protein and RNA level to show whether it's there, it's not there. And there's some suggestion there are other cofactors and other enzymes that can do that. One that's probably implicated in the brain is furin, a serine protease that is pretty widely expressed in brain blood vessels. It's been a target in stroke in some preclinical models. So to me, that one makes the most sense, although, even plasmin can cleave the S protein.
Yeah, I guess my other question was, I guess there's a lot of cell death happening because of the virus. Could some of the stroke be because of the sort of circulating cell death and sort of debris in the microvasculature?
Yeah, the most interesting data I think about this is this concept of something I just learned about this year, which was something called neutrophil extracellular traps. They're called nets, and they're small accumulations of neutrophils. They essentially 2 or 3 neutrophils trap the pathogen, and there's some fibrin in there, and so but it has a measurable size, and it itself can occlude smaller blood vessels. And it's very, as you can imagine, very sticky as well. So these nets may be an important contributor to the cerebrovascular pathology. That's not been well studied yet, but they're difficult to track and visualize. So,
Okay, that's great.
So I know many of you really want a career in medicine. So if you don't have oh, there's another question. I was just going to say, feel free to ask Dr. Hinman about, what he did in undergrad to get into medical school if you're interested as well. But we have Andrew's hand up. So Andrew, go ahead and unmute yourself.
Hi, Dr. Hinman. I just want to thank you, first of all, for your research in this ongoing health crisis. But in terms of the new variants that have been discovered, such as the one in the UK and South Africa, which have mutations in their spike proteins, would you predict, or I don't know if there's been any research on it, but would that make it more effective at binding to ACE2 in humans?
yeah, it might. I think the most concerning thing about the variants is the other important aspect of the spike protein is it somehow regulates transmissibility, which the way I think about that is that again, I'm not a virologist, but the way I've understood it is that the integrity of the spike protein allows the virus to exist outside the body. It acts sort of like a coat. And so if you mutated it in a way that makes the coat more stable, the virus can stay airborne or in a droplet for longer, increasing the transmissibility. I think the data about whether it's doing something to the severity of the disease is unclear. And the most interesting way a virologist put this to me was that SARS is a little bit like a hit and run virus. Some viruses get into a cell and they want to stay there, and they stay and they replicate and spit out virus constantly. HIV is a good example of that. But the SARS viruses, like many of the respiratory viruses, they attack cells, they replicate a little bit, and then they move on. And so that's, I think, we're probably going to see, if there are mutations that are allowing the virus to spread more easily, I'd be surprised if they also are allowing it to be more severe, because those things in viral biology, those things are incongruous, right? You can't have a virus that kills people and spreads rapidly. It has to do some combination of those two. Unfortunately, I think COVID-19 found the sweet spot, and that's bad for humanity. But I don't know that these viruses are necessarily going to do, the mutations, the variants are going to do something, cause a worse form of the disease or more complications. But I don't know that we know. I don't think we have a good handle on that right now.
Well, thanks so much, Jason, for that talk. That was really informative. I think we can open it now to our career panel, and Jason, you're welcome to stay on for that. But we usually have a few people that are going to. You can ask questions on any aspect. You can ask questions about, getting into undergrad. You can ask questions about medical school. You can ask questions about UCLA. I think Nanthia and Vidya can probably introduce this career panel a little bit better than I can, but I'm not quite sure how we're.
Yeah,